welcome everyone to today's lecture in relation to plant physiology and metabolism course. The topic of today's discussion is nitrate assimilation in plants. As you know that the nitrogen is an essential nutrient for plant growth and development, and one of its primary sources is nitrate. Nitrate assimilation is a vital process through which plants convert nitrate into organic nitrogen compounds that they can utilize for various metabolic processes. The first we will discuss about nitrate uptake. The ammonium ions have a brief existence in soil as they are rapidly converted into nitrate ions by nitrifying bacteria. Nitrate is the predominant form of nitrogen absorbed by plants from the soil. Moreover, nitrate is taken up by the epidermal and cortical cells of roots from the soil solution, although foliar application of fertilizers can also lead to uptake from leaves. Nitrate concentrations in the soil can range from micromolar to millimolar levels. Soil nitrate can originate from the nitrification of organic compounds or from fertilizer applications. Despite the energy expenditure required for nitrate reduction to ammonium within plants, nitrate remains the preferred nitrogen source. This preference stems from nitrate's ability to be safely stored in vacuoles at concentrations ranging from 20 to 70 millimeter or transported to leaves via the xylem, where it can be stored or metabolized. When plants are supplied with ammonium nitrate fertilizer following nitrogen deficiency, the accumulation of glutamine within cells downregulates the ammonium ion transporter AMT1. Upon entry into epidermal cortical or endodermal cells, nitrate transport into root cells is facilitated by two classes of symporters located in the plasma membrane and tonoplast, along with an antiporter exclusively localized in the tonoplast. To cope with varying nitrate concentrations in soil and tissues, plant cells possess nitrate transporter with different affinities for nitrate. As you can look that nitrate transporters are categorized into high affinity transporter system, in short it is called as HATS, and the low affinity transporter system that called as a LATS, based on their affinity for nitrate. These transporters operate within micromolar to millimolar nitrate concentrations. Heads functions effectively at nitrate concentrations ranging from 0.2 to 0.5 millimole, while LUTs operate optimally above 0.5 millimole nitrate concentration and do not exhibit saturation effects. Within the category of heads, there are two subtypes which constitutive are chats and inducible are heads. Constitutive heads are constantly expressed even in the absence of nitrate, whereas inducible heads are only expressed when nitrate is present. The expression patterns of these transporters vary across different tissues and developmental stages. Structurally, nitrate transporters consist of 12 membrane domains organized into two sets of 6 helices each. These helices are separated by a hydrophilic domain that extends into the cytosol. As illustrated in the figure, the uptake of nitrate is facilitated by symporters located in the plasma membrane operating in conjugation with the proton gradient. This process functions through a 2 hydrogen ion or nitrate symport mechanism. The establishment of the hydrogen ion gradient across the plasma membrane is composed of ATPase mediated hydrogen ion pumps. The movement of hydrogen ion along their gradient is coordinated with the uptake of nitrate. To regulate the concentration of nitrate within the cytosol, excess nitrate is either translocated through the xylem or stored in vacuoles. Within the tonoplast, a specific class of nitrate transporters belongs to the chloride channel family. Moreover, chloride channel family proteins can serve as either proton-gated anion channels or 2-nitrate hydrogen ion antiporters, facilitating the transport of nitrate across the tonoplast membrane. CLC protein transporter may exhibit a dual affinity system. When phosphorylated at threonine, they operate its high affinity transporter, whereas dephosphorylation converts them into the low affinity transporter. The maintenance of nitrate homeostasis within the cytosol is governed by a balance between nitrate uptake, efflux, reduction, and storage in vacuoles. Nitrate reduction takes place throughout the various parts of the plants. Although certain species like white clover, Trifolium repens, or cranberry, that is Vaccinum macrocarpon, primarily undergo nitrate reduction in their roots, while in others like cocklebur, that is xanthan species, this process occurs exclusively in leaves. The distribution of nitrate reductase activity within the plant is also influenced by the availability of nitrate concentrations. In temperate legumes, at low nitrate concentrations up to 1 millimole, nitrate is directly assimilated in the roots. However, when the nitrate concentration exceeds 1 millimole, its transportation to the shoot becomes notable, facilitated by nitrate transport via xylem sac. Conversely, in tropical and subtropical cereals and grain legumes, nitrate assimilation primarily occurs in the shoot irrespective of the external nitrate concentration. Whereas in maize, nitrate reduction predominantly occurs in mesophyll cells rather than bundle sheath cells. As mesophyll cells possesses a higher capacity for generating the necessary reductant, that is NADPH, which is essential for nitrate reduction. As you can see in the figure, nitrate assimilation involves two distinct steps. First, nitrate is reduced to nitrite, which takes place in the cytosol of the cells and facilitated by the enzyme nitrate reductase. Subsequently, it has been transported to the plastid where nitrite is reduced to ammonia, where the enzyme nitrate reductase or NIR is localized. The reduction of nitrate to nitrite involves the transfer of 2 electrons from an donor to nitrate while consuming 1 proton. This reduction is coupled with the oxidation of either NADH or NADPH, which typically serves as the electron donor in most plants. Land plants and algae primarily utilize NADH, while fungi utilizes NADPH for this purpose. However, certain forms of NR in land plants and algae have the flexibility to use either NADH or NADPH. This reduction reaction takes place in the cytosol of the cell. The active form of NR exists as a homodimer with the molecular weight ranging from about 100 to 114 kilodalton. Each monomeric unit comprises approximately 1000 amino acid and is structured into 3 domains associated with cofactors. These cofactors include the molybdenum cofactor, heme, and FAD. The N-terminal region of each monomeric units bind to molybdenum cobalt while the C-terminal region binds to FAD. The central region of each polypeptide unit is involved in complexing with heme I. The molybdenum co-center consists of molybdenum complexed with organic molecule, that is terene, it is here. As you look from the figure that nitrate reductase dimer consisting of 3 binding domains whose polypeptide sequences are similar in eukaryotes, molybdenum complex, heme and FAD. The heme F component include the cytochrome B5. The FAD binding domain comprises approximately 260 to 265 amino acids. The central heme domain consists of 75 to 80 residues, and the molybdenum cobalt binding domain of the enzyme protein is relatively large, consisting of 360 to 370 amino acids. Each domain of the enzyme functions as an autonomous unit and is believed to belong to a distinct protein family. These 3 domains within each monomeric unit of the homodimeric enzymes are connected by hinge region, that is HI or HII. HI region is located between the molybdenum cobalt center and the heme domain of the polypeptide, while the HII region is positioned between the heme domain and the domain complex with FAD. The HI region is known to play a regulatory role in maintaining the enzyme activity. NR activity is tightly controlled with the regulation occurring at both the transcriptional and post-translational level. Transcriptional regulations, which involve the activation or inhibition of gene NIA, encoding NR is a time-consuming process, taking hours or days to manifest. Conversely, post-translational regulation allows for rapid adjustments in enzyme activity within minutes. Notably, NR is identified as a substrate-inducible enzyme, with nitrate serving as a key inducer. Nitrate not only stimulates NR synthesis but also influences the transcription of numerous other genes. Specifically, genes responsible for nitrate transporters, pyridoxine, enzymes involved in the pentose phosphate pathway, and the PEP carboxylase are upregulated in response to nitrate. Conversely, genes involved in carbohydrate storage are typically downregulated, while those involved in amino acid and organic acid synthesis are upregulated. This regulatory mechanism underscores the pivotal role of NR in redirecting carbon resources from carbohydrate storage towards amino acid synthesis. In Arabidopsis, nitrate induces the synthesis of the molybdenum cobalt factor and regulates the synthesis of nitrite reductase, ensuring that nitrite generated during nitrate reduction is promptly assimilated to prevent toxicity. Nitrate reductase activity is tightly regulated with potential regulation covering at the transcriptional level of the NII gene, which encodes NR. The transcriptional regulation is time consuming while post-translational regulation occurs rapidly. Tang and Wu's 1957 demonstrated that nitrate reductase is inducible by its substrate nitrate, which not only stimulate NR synthesis but also influences the transcription of other genes. These include genes related to nitrate transporter, ferredoxin, enzymes in the pentose phosphate pathway, and PEP carboxylase, which are upregulated. Conversely, genes involved in carbohydrate storage are downregulated, while those associated with amino acids and organic acid synthesis are upregulated, illustrating the nitrate reductase role in directing carbon from carbohydrate storage towards amino acid synthesis. In Arabidopsis, nitrate induces the synthesis of molybdenum cobalt factor and regulates the synthesis of nitrite reductase to prevent the accumulation of toxic nitrite resulting from nitrate reduction. Rapid assimilation of nitrite is crucial due to its toxicity to the cell. Light promotes the transcription of the NI gene, which encodes nitrate reductase. In certain cases, sucrose can substitute for light to fulfill this requirement. In the dark, additionally, besides its role in generating reductants and photosynthase, light may also impact signaling pathway involved in photomorphogenesis. Following transcription initiation, messenger RNA levels of nitrate reductase exhibit diurnal fluctuations, possibly influenced by phytochrome activity. At the end of the dark period, there is a decrease in nitrate reductase mRNA, possibly due to the accumulation of glutamine, which inhibits NR gene transcription. Post-translational regulation of nitrate reductase occurs rapidly within minutes through phosphorylation and dephosphorylation of the enzyme protein. Phosphorylation renders the enzyme inactive, while dephosphorylation activates it. ATP-dependent phosphorylation targets a specific serine residue in the HI region catalyzed by calcium-dependent NR kinase. The activity of NR kinase is inhibited by light, triose phosphatase, and hexose phosphatase. Post-translational regulation of enzyme activity involves the interaction with 14-3 regulatory protein known for their inactivation role. These proteins derive their name from their distinct separation pattern during purification. Binding of 14-3 proteins to phosphorylated serine residues in the HI region of the enzyme disrupts electron transfer between the heme and molybdenum cobalt cofactors. The formation of NR 14-3 complexes is favored in the dark due to elevated magnesium levels, but is dismantled in light when magnesium concentration decreases. This regulatory process is also facilitated by AMP and IP. The NR1433 complex is susceptible to degradation by cytosolic proteases. Dephosphorylation of NR prevents 1433 proteins from binding to this particular enzyme, with dephosphorylation catalyzes by protein phosphatases. AMP and IP further promote the dephosphorylation processes. This reversible regulation of enzyme activity occurs rapidly within minutes in response to changes in light, darkness, or stress condition. As the figure illustrates that the regulation of nitrate reductase activity when inhibition is represented by the red color and green circle basically represent the promoting effect. Moreover, the factors like nitrate, light, and carbohydrates impact the regulation of nitrate reductase both at the transcription and translation stages. In barley seedlings, the detection of nitrate reductase mRNA occurred around 40 minutes after nitrate addition, with the peak levels reached within 3 hours, as you can see in the figure. However, while mRNA accumulation was swift, nitrate reductase activity showed a gradual, steady increase over time. This indicates that the synthesis of nitrate reductase protein is dependent on the presence of nitrate reductase mRNA. Nitrite is a reactive and potentially harmful molecule within plant cells. Hence, immediate action is taken to transport it away from the cytosol. This transportation occurs into specialized organelles such as chloroplast in leaves and plastids in roots. Once inside these organelles, nitrite undergoes a reduction reaction mediated by the enzyme nitrite reductase. This enzymatic conversion transforms nitrite into ammonium as shown in the reaction, that is a less toxic and more readily utilizable form of nitrogen for the plant. The reduction processes involve the transfer of 6 electrons, a crucial step in detoxifying nitrite and facilitating its incorporation into metabolic pathway in the reaction. Here, pyridoxine represent by the FD with the subscription red and ox indicate the reduced and oxidized state respectively. Pyridoxines serve as an electron carrier in this reaction, shutting electrons to and from the nitrite reductase enzyme. Through this process, plants efficiently manage and detoxifying nitrite, ensuring the power or proper utilization of nitrogen resources for growth and development. NIR is an enzyme represent constitutively in plants, although in some species its synthesis can be induced by the presence of nitrate encoded within the nuclear genome. NIR is initially transcribed into mRNA, which is subsequently translated by cytosolic ribosomes. Cytosolic NIR proteins contain a transit polypeptide sequence at their N-terminal region, which guides them to the plastid surface where they gain entry upon entry. This transit sequence is cleaved as the polypeptide traverses the plastid membranes. As illustrated in figure, NIR is composed of a single polypeptide with a molecular weight ranging from approximately 60 to 70 kilodalton. It comprises two distinct domains: the ferredoxin binding domain located at the N-terminal region and another domain situated at the C-terminal end. The terminal domain harbors 3 lysine residue critical for ferredoxin binding. Conversely, the C-terminal domain contains a single cluster of 4-iron 4 and 4-seroheme prosthetic group, which are closely situated and linked by sulfur ligands as displayed in the figure. 4 cysteine molecules arranged in two clusters on the polypeptide provide bridging ligand and sulfur ligands cluster. Seroheme serve as the binding site for nitrite. Moreover, in plants, the reductant used for NIR activity is reduced ferredoxin. However, in bacteria and fungi, NIR utilizes NADPH as the reductant instead. This figure shows the process of coupling of photosynthetic electron flow to the reduction of nitrite by nitrite reductase. Several intricate molecular interactions occur within the chloroplast. Firstly, during the light-dependent reaction of photosynthesis, light energy is absorbed by chlorophyll molecule, initiating the flow of electron through the photosynthetic electron transport chain. As a result, electrons are transferred to ferredoxin, a key electron carrier protein located within the chloroplast. These electrons are subsequently transferred from ferredoxin to nitrite reductase, as you can see, which contains two crucial prosthetic groups that are 4-iron 4s cluster and heme group. The 4-iron 4s cluster acts as an electron carrier, accepting electron from ferredoxin and delivering them to the active site of the enzyme. Meanwhile, the heme group facilitates the transfer of these electrons to the nitrite molecule, initiating its reduction to ammonium through the coordinated action of reduction and the prosthetic group within nitrite reductase. The potentially toxic nitrite molecule is converted into the less harmful ammonium ion. This conversion is essential for the assimilation of nitrogen into organic molecules, ultimately supporting the growth and development of the plant. Overall, the coupling of photosynthetic electron flow to nitrite reduction by nitrite reductase represents a vital biochemical pathway within the chloroplast, ensuring the efficient utilization of nitrogen resources and contributing to the overall productivity and health of the plant. In conclusion, it can be said that nitrate assimilation is fundamental process that allows plant to utilize nitrogen efficiently for growth and development. Understanding the mechanism and regulation of nitrate assimilation is essential for improving crop productivity and sustainable agriculture practices. Thank you.